This is topic number 2 of Unit 4 IAL Edexcel Biology. In this topic, we're going to be working on efficiency of food chains. We'll talk about something called productivity of food chains, which means how much energy is wasted and how much energy has been used along a food chain. We're going to also be doing some calculations and we'll do quick revision of the terms that you've studied in Unit 2. The first thing we're going to be working on here is the efficiency of plants, producers in a food chain. Now, look at this diagram. What do we see here? We could see that the leaf of a plant is receiving that much energy in kilojoules. Now, out of 100 kilojoules, let's say, there's 50% of this that's been reflected or comes in the wrong wavelength. So plants don't make use of all of the energy that comes from the sunlight because a lot of this energy comes in the wrong wavelength, meaning that there isn't the actual wavelength of light that is absorbed by chlorophyll molecules inside the chloroplast. There's another factor that reduces the efficiency of plants or reduces their productivity, which is the fact that a lot of light is reflected. Like leaves are quite shiny, so some of the light energy has been reflected. Some of the light energy penetrates without hitting any of the chlorophyll molecules. So as you can see here, out of this 100 kilojoules, how much energy we've been left with? We've been left just with 40%. And that's quite small amount of the original energy that we've received from this sunlight. Now, look at this 40 here. This 40 won't be transferred to the next trophic level. So the herbivore that would eat this particular plant here wouldn't get that 40% because the plant itself still needs to stay alive. Plants do respire. So you can see the mitochondria here. These 40% of energy that are going to be used by the plant itself to respire. So very small fraction. Actually, there'll be only left 10% to be passed from this original 100% of energy to be passed to the next trophic level or to the herbivore that is eating this plant. So we're going to list this amount of energy that is used by the plant. We're going to list it in the form of a diagram. Now take a look here. This is the original 100% of kilojoules that's been received by the plant. We've seen earlier there's three main factors that reduce that amount of energy. So we have reflection, we have the color of light or the wavelength. We also have the fact that light penetrates without hitting the chlorophyll molecules. So we were left here with just 40 of the original 100. Now this 40 is what we call the GPP or gross primary product. Let me write it here for you. So gross primary productivity. All right. Now, this GPP or gross primary productivity is the energy that is available for the plant itself. So the plant will make use of this GPP to stay alive, to respire, to grow, and to do all of the activities that the plant would do to remain alive, like active transport. But due to respiration and this waste of energy that's been done by the plant itself, there will be smaller amount of energy. That's what we call MPP. So MPP is always going to be less than GPP. MPP stands for net primary productivity. So which is always less because it's GPP, basically it's GPP minus the energy has been wasted by respiration. All right. Now, this MPP is what's going to be available because this MPP is like the starch or the cellulose that's been made by the plant and is available for organisms. So like it would be available for decomposers later on, and will be available for the herbivores. So basically, all of the biomass that's been used by the plant to grow and to make materials that are edible by the herbivores or the decomposers is what we call the MPP. We're going to combine all of the information that we've learned so far into one neat formula. So we're still talking about the productivity in plants. Now, let's try first to do a quick recap on the new terms that we've learned today. Now, first, it's the gross primary productivity. Gross primary productivity is the total amount of energy that is converted to chemical energy due to photosynthesis. Out of this GPP, or gross primary productivity, there is always energy that is wasted by the plant to stay alive by the mean of respiration. That's the release of energy. And what is left now is net primary productivity. So net primary productivity is the energy that is left after the loss due to respiration. So the formula is going to be like following: MPP equals GPP minus respiration, right? This is quite important formula. It's not only about the calculation. Calculation is quite simple, but it's useful to list this formula while you're writing a long answer related to productivity and efficiency of food chains in general. Now, MPP is quite different from one habitat to another. It really depends on how much is the density of plants in this particular habitat. So, for instance, if this was like a desert habitat, now what would you expect there? You would expect the density of plants, or the number of plants per unit area, you would expect it to be small. So for that reason, such a habitat would have low gross primary productivity and low net primary productivity. This is opposite to what would you expect to have in a forest where the density of plant is quite high. Now, what's really important here is the units. So what are the units we use for MPP or GPP? It's usually units of energy, kilojoules, per units of area per unit of time. So it's always this particular unit that we use. Meters could be kilometers, but it's always years and it's always kilojoules. One way to measure productivity rather than kilojoules is quite difficult to measure the energy that's available in a tree or something by measuring how much energy it is, but we have an alternative. The alternative here is to measure dry mass. Now, dry mass represents how much energy or calories stored in a tissue or a food sample. How would you measure the energy stored, or how would you measure dry mass? Usually you have to take a leaf sample. So I take a leaf sample of this tree that I want to study. What do I do here? I would dry it up. So I'll place it in an oven. And when it's placed in an oven for like 24 hours or so, it dries up. Why am I drying it up? I want to dry it up because I'm interested in the living tissues. I'm interested in biomass. I'm not interested in water here. So the dry mass would be a good indication of how much energy there is, right? So you could do this for a tree. You take a few leaves, like you actually need a large number of leaves, like take 100 leaves. Do how much energy is there at the beginning of the year, and do the same thing at the end of the year. That would give you a rough calculation of how much energy there is in this particular tree or in this habitat. Now, here I want to remind you of something you've probably done in your IGCSE. This talks about the efficiency of food chains in general, not only about plants. Now, we've seen the first part here. We've seen that plants, the MPP for a plant is around 10% of what is received by the sun. Now, still, we have a lot of waste of energy. So energy is continuously lost along the food chain. This is why we have very little energy left at the end of the food chain. So that's why you wouldn't expect to have a food chain that is more than 5 levels, because very small energy is left at the end of the food chain. So for instance, why do we lose a lot of energy among those consumers here? There's many reasons here. Like, consumers continuously lose energy because consumers usually are animals. They lose energy due to movement. That's a lot of waste of energy. Respiration, just like plants. And excretion. So those are 3 major processes that cause loss of energy. So basically, there is a rule here in ecology. It says like only 10% of what is available for this grasshopper, only 10% will pass along to the next trophic level, and only 10% of the 10% will be passed along to the next trophic level. There's also something about animals, which is like there's so many tissues in the body of the animal that are not edible, like the bones and the fur, the nails. So all of these are not edible, so that adds to this loss of energy in a food chain. Usually, as we said earlier, we call the energy available in a plant, it's like primary productivity. The energy available in an animal, we call it secondary productivity. So secondary productivity is like how much energy is available in this mouse to be presented to the next trophic level, to the snake here. There's one common calculation that you may be asked about, which is like efficiency. How would you work out efficiency in a food chain at any trophic level? So here's an example. Look what it says. It says like, if grass receives 1 million kilojoules of energy from the sun, and it uses 20,000 of energy for growth. When it is said for growth, this means this energy available for the next trophic level. So how much is the efficiency here? What would we do? You would take 20,000 and you divide it by 1,000,000. That's 1,000,000. And just like you're doing a percentage here. So that's the efficiency of this particular food chain. So that's around 2%. We'd always expect this efficiency to be less than 15 or 10%. We're going to go back to the syllabus just to check how far we are here. Now, look where we've reached. We've reached the last part of this topic, ecology. So right now we're going to be talking about the ecological terms like population, community, habitat, and ecosystem. So let's start with the population. What's a population? Now, take a look at those horses. This is a population. A population is usually a group of organisms that they have to have 3 things in common. What are the 3 things in common that they have to have? First, they're going to have to be members of the same species. That's a must. They're going to have to live in the same habitat. And they're going to have to be at the same time, right? So it doesn't make sense if the horses that lived 1,000 years back, they died out, and horses that live in right now, so those don't belong to the same population. All right, so that's one particular term. Now, there's another term here. There's another term that's quite important, which is a species. Like, how would you determine that 2 animals belong to the same species, or just 2 organisms in general? How would you determine that they belong to the same species? Now, for instance, a horse and a donkey, those don't belong to the same species. We know that, but they do get offspring. They get a mule if they breed together, but nonetheless, they are members of different species. How will you determine that two animals in general, how will you determine that they belong to a different species? What would you do here? The offspring, the mule that we've produced here, the mule has to be able to breed. It has to be fertile. So since the mule is infertile, like mule and a mule cannot breed together, so since they cannot have offspring, then the parents, the donkey and the horse, then the parents belong to different species. So again, the definition of species is quite simple. What would you say? You say they're animals, or just organisms in general. They have to breed. Not only this, but the offspring have to be fertile. So they have breed to produce fertile, that's a must, offspring. So organisms that breed to produce fertile offspring. We're still doing a revision of the important terms here. Now we have another term here, which is ecosystem. Like, what does an ecosystem stand for? An ecosystem is like a group of organisms. It could be animals, it could be plants. Those organisms, they're gonna have to interact. How would the organisms interact with each other? Organisms interact with each other by like herbivory, predator-prey relationships. So all of those relationships between organisms, they're usually feeding relationships. That's one important factor of an ecosystem. So ecosystems represents the relationships between organisms among each other. But ecosystem also includes the non-living habitat. So to define an ecosystem, what would you say? We'd say like it's interactions between living things together and their non-living habitat. We usually have a term for living things, interactions of living things. We call this biotic factor. So what's a biotic factor? A biotic factor is just any factor related to living things, like predator, prey, like disease, bacteria, and all of these things. What about abiotic factor, or the non-living habitat? This is like temperature, sunlight, water, soil, minerals. All these are abiotic factors. So an ecosystem is both the biotic and the abiotic factors of their habitat and their interactions. Biotic interactions include two types. So there are two types of biotic interactions, or interactions of living things. These are usually, we call them intraspecific. When it's intraspecific, this means it's interactions between members of the same species. So take a look at those two animals. These are usually males, and they are competing to breed with a female. The one that is better fit, the one that is more adapted, is the one that would be able to pass its DNA to its offspring. We also have another form of biotic interactions, which call it interspecific. Interspecific is like interactions among members of different species. This is usually interactions for resources, for habitat, for shelter, food, prey, etc. Now, these interactions, they're always putting what we call selection pressure. When you study evolution the next time, we'll talk a lot about selection pressure. But nonetheless, for the time being, selection pressure is what keeps the population size always in check. It keeps the population number, or the number of organisms of any species, keeps it usually stable. Another important ecological term is the term niche. Now, what's a niche? A niche is the function or the role of an organism in its habitat. Usually, every organism within its habitat has its own function. For instance, Think of this bird, the blue bird right here. What would be the niche of this blue bird? It would represent its feeding relationship, like what does it eat and who eats this particular bird. Same would apply for this pink bird there on the left. So it would also has its own niche. Nonetheless, in many cases, niches overlaps. Like when you have an overlap of the niches, that usually happens all the time, but usually organisms try to keep a distance. They try to minimize the competition. But when conditions change, like when you have drought or when you have less food, then the competition starts to increase because there's a limited supply of food. At that time, should have said, now at that time, the organism that is better adapted is the one that would win this competition, and it usually excludes the less adapted organism. Nonetheless, the normal case is that you have division of niches within the habitat. So always habitats have, like here, every bird has its own niche because it feeds on different type of food than the others. This ensures that all of these birds are actually surviving and the population size remains stable. I would like to return back to the concept of niche one more time just because of its importance here. So like, what if you've been asked to describe the niche of a specific organism? That's a very common question. How would you answer such a question? So for instance, someone asked me like, what is the niche of this particular bird here? All I have to do here is to list the feeding relationships that this bird has. So for instance, I would say that this bird provides energy to the larger bird there on the tree. It controls the size, or it feeds on those worms, so it controls the population size of those worms. So that would be the role of this bird in this habitat. Usually when it's a tree or a plant in general, you could add more to this because plants provide energy for the entire ecosystem. So I would say if I was asked for the niche of this particular tree here, I would say it provides food for the birds, worms, and all of the other organisms. It also provides like a habitat for all of these organisms. So basically all we have to do to answer a question related to a niche is just to list whatever information you've been given in the question itself about these organisms. This diagram helps us to understand how populations remain stable in an ecosystem. So think about it this way. When the population size is small, like if you have few organisms within this population, then there will be less competition. What would that lead to? Since we have now plenty of food, few individuals, then we will have an increase in the population size. But as the population size increases, there's something that's going to happen. There'll be more competition because there'll be less amount of food. Now, as the competition increases, then what will happen? That will lead to a decrease in the population size. And then we return back to where we've started. So you can see here, the population will remain stable as long as there is no external interference. So usually all populations within an ecosystem are stable unless there's like a natural disaster or human interference. I wanted to pause the video for a moment here and try like to do a recap about these terms that we've learned so far. So first is ecosystem. Now ecosystem includes all living and non-living organisms in a habitat. The non-living part is what we call a habitat. Community, this is different species. So community is members of different species within the habitat. But if they were members of one species, then it is a population. Finally, a niche is the role of an organism within its habitat. We're now getting to topic number 3. This one talks about sampling techniques. So here we're going to be talking about how do we study habitats? How would we determine the population size of particular species within the habitat? So usually we use tools to study the habitat. So we use something known as quadrats. We also use something known as transects. So basically I will try in here to show you how do we use those tools to study population size and density of plant, for instance, in a habitat. Before we get into details, into practical details about the quadrats and the transects, let's just get familiar with these two terms. So one is abundance and the second is distribution. What does abundance stand for? Abundance is basically the number of organisms in a habitat. So when I say like this tree here in this particular habitat, this tree, tree A, is highly abundant in this habitat, but is less abundant in this first habitat. So basically abundance is a count of the number of one particular organism within its habitat. Another term here is distribution. Now, distribution answers the question of where. Abundance is how many. All right, so when I say how many, that means it's the abundance. When I say where is this organism found, that means it's distribution. So distribution is where is this particular organism is found, how is this rabbit or how is this tree distributed along a particular habitat. To study abundance, we use quadrats. What's a quadrat? Now, a quadrat is just like a frame where you have smaller frames or grid. This helps us a lot because it makes our counting much easier. Usually quadrats are used to count plants or animals that don't move much, because simply you drop it on the ground and you start the counting and you count like how many of these organisms are found. But you can also do something very useful. You could find what we call density or percentage cover. Percentage cover or density is like how much of this area of the quadrat is covered by one particular species. Usually the size of the quadrat is fixed, is usually 1 by 1 meter square. So this would be helpful because if this area is being covered, I can simply find the percentage cover of one plant in this habitat. So again, to find a density or percentage cover, which is like the same thing, you would find the number of individuals found divided by the area. When we use the quadrats, we have two systems. We have two ways to do the abundance or percentage cover. One way we do it by means of what we call random sampling. Like, what is random sampling? So random sampling stands for like you get to the place where you want to study and you do random walks and you drop your quadrats every now and then. This is usually done when you have a large habitat and you cannot cover the entire area. So it's not like very accurate method, but if you've been fair, if you've dropped your quadrats at quite distances and you've been like not biased, basically you took representative areas of this habitat, then that would be just good enough. It would be very helpful to do like comparisons. If you have two habitats and you would want to compare the distribution of let's say these flowers. You want to study the percentage cover of these flowers in these two habitats, then simply you study these two habitats randomly and you find how much is the percentage cover here, and that would be helpful to do this comparison. So here's an example of me doing random sampling. So let's say I want to study the percentage cover of those flowers in this habitat. So what will I do? This is my quadrat. So I have this here, and then I count how many of these flowers are there. And then I repeat this method. So I take my quadrat now again, and I do this somewhere else. So just move the quadrat itself, and you do random sampling of those flowers in the first area. Now let's say I want to compare the number of those flowers to another habitat where there's like some factor that I'm interested in. So let's say I have some factory here and I want to study the effect of some chemicals on those flowers. So what I do, I go there at the same time of the day. So I have to be fair. I have to control variables such as the weather, such as the light intensity, and also best thing to do is to visit the area at the same time of the day and do the percentage cover and do some comparison between those two habitats. Remember earlier we've indicated there are two terms that we're interested in. One is distribution and the other is percentage cover or abundance. Now, to study distribution, we have a very neat method, which is the transect. What's a transect? A transect is simply a long tape where it's the measurements, and I place this transect along my habitat, and I see how many, or where, I should have said, where are these plants are found. So like I place this along my habitat, and I see what parts or at what distance these plants are touching my transect. So let's say I was studying this particular plant, then I will find that it's not found here, the beginning is not found at the end, is mostly distributed somewhere here in the middle. Transects, as I said, they don't show you the percentage cover or the density, but they show you the distribution. But you could do a combination of the quadrat and the transect. So you place your transect, and then you bring your quadrat, and you put this quadrat along your transect. So I put it here, put it here, put it here, put it here, and that would be 2-in-1, because this means I've done the distribution because I know where these plants are found, and I've done the percentage cover because I could, based on my quadrat, I could find how many plants are found there. Here are some examples that I found of the use of transects. So this gentleman here, look what is he doing? He has placed his transects along this habitat, and he's studying the distribution of one particular plant. So basically, wherever this plant touches his transect, he would be indicating this on his tally chart, on his graph, and that would show him the distribution. Same thing could be done here under the sea, studying like certain corals here. Again, the transect is placed, it has like divisions, and he could find where are these particular corals found. Just like what I told you earlier, you could use the transects, like you could place more than one transect just to have the quadrat at the right position, and using this system here, you have indication of where, and you have, using your quadrat, you have an indication of how many plants or snails are found within this habitat. This method has a name. We call it usually a belt transect. Belt transect meaning that you have a quadrat and a transect. And as you can see here, this is what we call systemic sampling, because you're not doing random quadrats everywhere in the habitat. No, you place your quadrats in a specific way based on a specific distance that is determined by your transect. So I found another example here. We could take a look. This gentleman here is placed at transect. He wants to study the distribution of dandelion, or any plant basically, along his garden here. So he placed this transect again all along, and he's placing his quadrats along this transect. And what he's doing here, simply, he has a table here of distance, meters, versus the number of plants or whatever organism he is interested in. And you can see here, we have a steady increase in the population size as we walk along this mini garden or habitat. Each method, the random or the systemic sampling, each one of those methods has its advantages and disadvantages. So both methods are quite useful to find percentage cover. However, systemic sampling could be sometimes impractical. When will it be impractical? That's when you have like very large habitats and it's impossible to study. It's quite time consuming as well, because you're gonna have to cover as much of the habitat as possible. But on the other hand, systemic sampling is fair because you're following a specific transect to study the habitat. Random sampling could be biased because it would be based on your preference, it's based on your comfort, so it's not going to be the very fair method of studying the habitat. And as I said, it's going to be less accurate than systemic sampling. Now, how would you make random sampling? How would you make, if you like to study big habitat, how would you make random sampling less biased? Usually we take like an aerial image of the area that we are to study, and then we draw like virtual grids on this area. Next thing now is that we make coordinates. We generate numbers using dice or an application, and then we, instead of being like based on our preference, now we're placing our quadrats based on random numbers that are generated randomly. So that would be the best way to study large habitats using this random sampling method. Here is an example of how random sampling could be done in a neat way. So first thing to do is to find the area size of the one you're studying. Then you generate a grid, like with coordinates. Later on, you're gonna generate random coordinates using an application. So for an example, so these are random numbers, random coordinates. After that, we do the percentage cover. So we place our quadrats here in a random way. We counted the population size in each position. You can see the numbers are quite close. Then we find the average or the mean. This is the mean number of plants per quadrat here. So instead of me doing this for the entire area, I multiply this, because each quadrat is 1 meter squared. I multiply this by the total area, 620, and that gives me the size of the population in the entire area.